Chromatography is a separation technique so named because it was originally used to separate pigments of different colours. Nowadays, chromatography is used by biologists to separate all sorts of mixtures into their component molecules. Chromatography can be very high-tech or very low-tech, but all types of chromatography involve a stationary phase, usually a solid, and a mobile phase, a fluid, which acts as a solvent for the mixture to be separated. The mobile phase moves across or through the stationary phase, and separation occurs because different components of the mixture have different affinities for the stationary and mobile phases. Therefore, components of the mixture move over or through the stationary phase at different speeds. Chromatography is a useful way to identify the components of a mixture or to purify a desired component from a mixture. Thin layer chromatography, TLC, is a simple form of chromatography used primarily to identify the components of a mixture. The stationary phase is a thin layer of silica spread on a glass or plastic sheet. Silica is a porous, highly polar, adsorbent material. The mixtures to be analysed are applied to the silica near the bottom of the plate. Then dried completely to fix them in position. The plate is placed in a jar with a lid. The mobile phase is a non-polar solvent such as butanol. The mobile phase forms a layer at the bottom of the jar. It does not reach up as far as the samples, or samples would get washed away. The solvent is gradually drawn up the plate by capillary action, and the samples, dissolved in the mobile phase, move upwards with the mobile phase. The different components of each sample have different affinities for the polar stationary phase. Polar molecules have a tendency to absorb onto the surface of the polar silica. This slows their progress, but does not cause them to stop moving completely. Non-polar molecules have little or no affinity for the silica, but do have affinity for the non-polar solvent and thus travel further up the plate. A way to remember this is like dissolves like. In a given space of time, the components of the samples will separate out according to their respective polarities. When the solvent front nears the top of the plate is taken out of the jar. The solvent front is marked with a pencil, and the plate is immediately dried so that the solutes precipitate onto the plate in their final positions. In the example shown here, you can see the different components of the samples because they are coloured. However, in most cases, the samples are colourless. To view the spots on the plate, a suitable stain must be applied. For amino acids, for example, ninhydrin stain is used. The distance travelled by each solute is expressed as a fraction of the distance travelled by the solvent front. This number is called the RF value, or retention factor. The lower the RF value, the greater the affinity of the solute for the stationary phase. Thin layer chromatography can be used to identify the components of a mixture, check the purity of a sample, or observe the progress of a chemical reaction over time. In affinity chromatography, purification of a particular component, the target molecule, is the goal. The stationary phase is a resin consisting of tiny beads of a porous substance such as saccharose. The beads are packed tightly in a glass column. The column has a filter at the bottom that prevents the solid beads from falling out but allows the liquid mobile phase to pass through. The mobile phase is simply an aqueous solution that the samples are dissolved in, for example phosphate buffered saline. There is a tap that allows us to control when the mobile phase can leave the column. The Sepharose beads are coated with a substance shown here in red that has a high affinity for the target molecule, but not for any of the other components of the mixture dissolved in the mobile phase. This means that when a sample is applied to the column, the target molecule, yellow star, will absorb onto the beads and be retained in the column while the rest of the sample passes straight through. A wash step is performed to remove any residual contaminants from the column. Having successfully isolated the target molecule from the original mixture, we must now collect it for further use. An elution buffer is added. This is a liquid that will cause the target molecule to be released from the Sepharose beads. The liquid that elutes from the column, the eluent, will contain the purified target molecule. Some practical considerations. When running an affinity chromatography column, there are two important points to keep in mind. Firstly, the Sepharose beads must be evenly packed. If there are gaps between the beads, the mobile phase and the target molecule dissolved in the mobile phase will not have adequate contact with the stationary phase, and the target molecules will not be retained on the beads. To avoid this problem, the beads must be packed down into the column in a uniform layer. This is achieved by allowing buffer to flow through the column, forcing the beads into the correct position. Secondly, the Sepharose beads must never be allowed to go dry. If the beads go dry, they will shrink, which will interfere with the tight packing of the column. Therefore, they must be kept under liquid at all times. The beads are added to the column in the form of a slurry, a semi-liquid suspension of beads in an aqueous buffer. During the subsequent washing steps, the liquid level must never go down below the top of the layer of the beads. The tap is closed as soon as the liquid level reaches the beads. Take-home points. TLC, thin layer chromatography, is a type of absorption chromatography, where the solute molecules separate according to their polarity and are identified on the basis of their final positions on the plate. Affinity chromatography is a powerful purification technique based on the selective but reversible adsorption of a biomolecule to its complementary binding partner. More details of a specific example of affinity chromatography are given in Part 2.